So thanks very much for the invite. I'm going to talk about genetics and phenotypic causation. And by that, I mean how one phenotype influences a downstream phenotype. And I'm going to discuss how genetics was fundamentally interested in phenotypic causation. Natural selection operates obviously through phenotype. There's not some magical way that genotype is going to influence fitness or outcomes, which does not go through some phenotypic pathway. And of course, many stories, some of them just so stories are put together about how phenotype is selected upon and how that drags genotype along with it. So, the peacock's tail is obviously, the genes influence that, and there will be some other non-genetic effects, stochastic and then possibly tangible environmental effects. And however the phenotype is influenced, the selection upon it will be the same, and then the phenotype somehow leads to greater fitness. So natural selection is through phenotype. How this could work was, of course, a mystery, and the eclipse of Darwinism, as it's been called, partly was due to the problem of blending inheritance. So Darwin's notion of pangenes, which was a sort of semi-Lamarckian notion, produced a model which would suggest that inheritance would blend. So the parental phenotypes, the parental genotypes, would produce a blended phenotype, and that with that sort of blending inheritance, Fleming-Jenkin famously demonstrated, but with some of his maths wrong, but anyway, demonstrated how how the selection would have to dissipate, how that couldn't produce selection, and that was his famous review. Of course, at the same time as Fleming-Jenkin was writing, in Moravia, Mendel was proposing the notion of particulate inheritance, but of course this was not widely known until its rediscovery in 1900, but provided a mechanism which would avoid the blending inheritance, which was so problematic in theory. Mendel's extraordinary paper doesn't pose two laws. We'll taught two laws. There are about 7 law-like statements that you can get through the paper. One of which would be dominance, for example, which is no longer considered a law. One of which is what is now thought of as a second law, or pretty close to what's now thought of the second law. The construction or invention of Mendel's laws was by Robert Heath Lott, an extraordinary but forgotten figure who wrote the recent progress in the study of variation, heredity, and evolution in 1906, which is a wonderful book. In that, he then put Mendel's laws into 1 and 2. The first being the complete segregation of the 2 allelomorphs in equal numbers of the germ cells of a heterozygote constitutes the first and most important section of the generalization known as Mendel's law. And then the second part of the law refers to the fact that, as a general rule, separate pairs of allelomorphs segregate quite independently of one another. Because Locke and others had already demonstrated linkage at that time, it had been demonstrated. So that's why he says as a general rule. And I think because of the salience of the biometrician versus sort of the Mendelian versus biometrician, or vice versa, fights, many of which, of course, UCL was responsible for. It's sort of forgotten that in Mendel's paper, he allowed for tiny phenotypic effects, and discussed examples where you couldn't actually see any sort of dominant, any phenotype. He allowed for that. In Bateson's and Saunders's report to the Evolution Committee in 1902, they allowed for that. George Udny Yule in 1902 had discussed that. Locke discussed it many times, that the very small Mendelian effects would become inseparable from a biometric model. And indeed, Pearson said, one of the first demonstrations of this, although he said that it couldn't actually account for example, heritability of height, because he assumed a dominant model. He didn't assume codominance. But Udny Yule assumed codominance and showed that the two were not really necessarily contradictory. But that doesn't stop the fights. And I'll come back to this at the end. I'm only going to relatively briefly mention Mendelian randomization at the end of the talk. But the principle of Mendelian randomization depends upon Mendel's two laws, and I'll come back to its on the laws later. It says live transcription. Yeah, just click there. There you go. That was even my extremely low technological ability could deal with that. Possibly the next important landmark is Wilhelm Johannsen's introduction of the term gene, but then also his introduction of the term phenotype and genotype, and attempting to separate the two. But as far as geneticists were concerned, people who were interested in how phenotype was transmitted, Johannsen was very clear that the actual nature of the gene didn't matter. He said it's not at present well enough established. This, however, is no consequence to the efficiency of research on heredity. It is enough that it can be asserted with certainty that such genes are available. So what they were didn't matter. And another important researcher who thought the same was Morgan. And Morgan was equally explicit about this, and people have used the term instrumental gene. That Morgan just thought of the gene as an instrument, not in the way we now think of instrumental variables. I'll get on to that. But he used it as an instrument to actually understand how genotype converted to phenotype, and how phenotype worked. It's the phenotypic chains that were then important. And reflecting back on this, when he collected his Nobel Prize, he said, what's the nature of the elements of heredity that Mendel postulated as purely theoretical units? What are genes? Now that we locate them in the chromosomes, are we justified in regarding them as material units, as chemical bodies of a higher order than molecules? Frankly, these are questions with which the working geneticist has not much concern, except now and then to speculate as to the nature of the postulated elements. There's no consensus of opinion amongst geneticists as to what the genes are, whether they are real or purely fictitious. They could be fictitious as long as it worked. Because on the level of which the genetic experiments lie, it doesn't make the slightest difference whether the gene is a hypothetical unit or whether the gene is a material particle. So it's very clear that Johannsen, Morgan, they thought of the genes were instruments. It was an instrumental gene, as the term has been used, for actually understanding what were phenotypic processes. So the next step in the UCL figure was R.A. Fisher. And of course, Fisher's famous paper, which built on the work of Udny Yule and John Brownlee and of Snow and of many others, with rather ungenerous footnoting, should we say, of these people who had contributed to this field, but with his what became known as the infinitesimal model and his complicated mathematical demonstrations of how this could work. So very interestingly, this paper, which actually introduced the notion of the analysis of variance, only uses the term once, which is actually in the contents. Nowhere in the paper does he actually say analysis of variance. He actually used the term analysis of variance again. So you had to be pretty perceptive to actually realize that this is the term that he introduced. He introduced that term in more detail in his eugenics review, popular version of this paper in 1919. This paper was almost published in full in the eugenics review, but Leonard Darwin paid for its publication in the Transactions of the Royal Society of Edinburgh. And the paper starts that several attempts have already been made to interpret the well-established results of biometry in accordance with the Mendelian scheme of inheritance. It is here attempted to ascertain the biometrical properties of a population of a more general type than has hitherto been examined. Inheritance in which follows this scheme. It is hoped that in this way it will be possible to make a more exact analysis of the causes of human variability. So Fisher's interest was in how this could actually apply to human variation, and of course this tied in with Fisher's interest in eugenics, because there had to be a substantial component that was genetic. If you could think that even in principle that was a possible aspiration. He chose height, which of course is about one of the most heritable factors there is in the human phenotype for most of the demonstration. Now, if hands up who's read Fisher's 1918, the original, the correlation, has read it. Hands up who actually got through it. Right. So the reason it's impossible to get through is it is just full of mistakes. It's just full of the most simple mistakes of moving from some simple ones of moving through the lines of maths, but it's just peppered with them throughout. I could never get through it. You would just stop at a certain point where you couldn't follow it. But I then discovered this 1966 from UCL, this 1966 commentary on it, which reprints the paper and then tells you all the places where there were mistakes. And the mistakes were, probably transcription errors. It was set desperately fast to get it into the Transactions of the Royal Society of Edinburgh once Darwin had paid them. And it was symbolically. It's an unread paper. I think a largely unread paper. But if you do want to read it, which the International Journal of Epidemiology, when I edited it, we put online, so you can find this together with a reprint of his 1919 paper, and it's linked to that. But you can read the original with but with the corrections to it, and it's then possible to read. Of course, so Fisher was decomposing phenotypic variants in the way that we now, would now call identifying heritability. But he didn't use the term heritability, wasn't used, but the concept was clearly that. At roughly the same time, Sewall Wright used the symbolic mathematical approach that he had developed of path analysis to decompose phenotypic variation the same way, but not with a focus on humans, explicitly not with a focus on humans. I think Wright is a genius, and one feels for Wright when you say in his biography, he'd read his father's maths books and learned to extract cube roots before entering school, a skill that he said brought him instant lasting unpopularity with the other students, which I'm sure is something we can all relate to. And so this is the famous, because beautiful diagram in about the third paper he did on path analysis, but the one that's always cited because of the diagram. And this is the decomposition of the influences on the guinea pig coat patterning, and he decomposed that, and he uses, what I really like about this is chance. He says, chance in the gamete formation. So that's why the chance, which gives you some ability to make causal inference in these chains down, as well as the fact that it's anchored by crosses. And he decomposed the variance, and he showed how the variance decomposition was completely different if you had highly inbred strains, because of course in highly inbred strains there's very little genetic variation, so most of the variance becomes intangible. In just ordinary crosses, still 58% of the variance is intangible. And I think this is sort of missed, I think, when people look at Wright's paper, and I think it's one of the most important lessons of the work that he did was that in many phenotypes, much of the variation is not something which you can actually put a name on. You can put a name to genetic variation, and that's real. And, you know, as Wright showed, the amount of genetic variation depends upon how much environmental variance there is, or how much genetic variance there is in the population. So it's a, you know, it's a shifting value, which is not a sort of set value. But I think his division into tangible and intangible environmental components is really key, and is quite close to the later notion of non-shared environment and shared environment. With shared environment, something you can sometimes actually say what the factors are. With non-shared environment, much of it is probably stochastic, either stochastic aspects of life or stochastic aspects of biology, mutations, epimutations, whatever. So, you know, he said that the differences must be due to irregularities in development, due to the intangible sort of causes to which the word chance is applied. And so he's saying that much of the phenotypic difference, what appears to be environmental, isn't the sorts of environmental differences which you can label in a useful way. The environmental differences that you could label in that way in his work were things like the age of the dam, the age of the mother, sometimes the weight, you know, factors like that. And so Wright, so what was controversial was that Wright was actually suggesting that you can use statistics to get at causation. So statistics were thought were sort of descriptive. Pearson did want to make causal claims. He was making causal claims to support his eugenicist notions that, for example, parental alcohol consumption didn't actually damage offspring. It was just that, you know, genetically predisposed people to drink had poorer quality, as the word he would use, offspring. So he wanted to make, he was making causal claims, but he used the language of just correlation, which becomes more and more perfect and allows you to predict what's going to happen. But Wright wanted to say that cause exists as a sort of ontological category. Some things do cause other things. And then that his path analysis, if the path analysis is appropriately anchored on something like crosses, or like, you know, genes, or like time, or some other factors that themselves can't be changed by what you think the outcome is. When you have some anchoring, you could actually make causal statements about the processes. And, you know, much of what he did with this was on anthropometry of rabbits, guinea pigs, lots of other small creatures that you could cross. As I say, he did quite a lot of work on antenatal environment, you know, maternal weight and age, and he looked at assortative mating through phenotype using these approaches. Very late in his life, some of his colleagues looked at obesity and lipoproteins, which is closer to the current day work. And then he looked at corn and hog variables, which I will get back to. That's another story. So Sewall Wright introduced this technique in his famous paper called Correlation and Causation in 1921. And then he was rather dismayed about the fact that it was then sort of applied and causal claims were made, or people just rubbished it and say you can't make causal inferences from data in this way. And his response to that was to say that it was, of course, realized that the concrete phenomenal actuality of the results was not provided by the analysis by path coefficients. So it wasn't your statistical analysis that allowed you to make causal claims. But that rests on the validity of the premises, i.e., what you know, on the which is the evidence for Mendelian heredity. So in most of his examples, it was crosses. It was essentially, you know, genotype, genetics, which provided that anchoring and allowed you to say that the phenotypic flows must be from that, not back. It must be a direction away from that. You could actually put a direction on the arrows. And he says that his paper began with a quotation from Easton Jones on the universality of Mendelian inheritance under sexual reproduction as the justification for the analysis. So making causal claims from his sorts of from these sorts of analyses required having such anchoring. He said that, the hypothesis that heredity is Mendelian may usually be used safely as information external to a system of correlations among relatives. And external information of a most precise sort is provided by the pedigree and by the practical universality of Mendelian heredity. So this was Wright's notion of phenotypic causation. But from William Provine's brilliant biography of Wright, because he interviewed Wright was alive when he wrote it. He said that after Wright published the method of path analysis, some researchers hoped that it was really a method for deducing causal relations. Wright repeatedly denied this, yet the hope did not die easily. The method was best applied to very definite causal relations, as in the case of Mendelian inheritance. But of course, later, really from the early 1970s on, path analysis was taken up enormously by the social sciences, and causal claims were made about everything that you could actually fit a path analysis to. And one of my favorite quotes of all was in the quite early days of that was realized to have moved away from what was doable. And Louis Gutmann said about the path analysis doesn't analyze non-genetic paths. Said genetics has but one modest framework for paths. In contrast, according to current journals, it means sort of social science journals largely, sociologists keep discovering new fundamental path frameworks every month, and sociological graduate students are required routinely to hand in as individual class exercises new discoveries equaling Gregor Mendel's. So this was the sort of disaster of the sort of technique. What Wright didn't want, which was the technique, was taken. And claims were made, causal claims were made on the basis of evidence that didn't justify it, because there was no anchoring of, so you couldn't put directions on the arrows. So a less known aspect of Wright's work is that he contributed to the development of what became known later as instrumental variables techniques. So these are were usually attributed to his father, who was the economist Philip Green Wright, who wrote a book in 1928 called The Tariff on Animal and Vegetable Oils. And in that book, or in one of the appendices of that book, he introduced the method of additional or external factors, which was basically saying when something happens, or cannot be influenced by what then is downstream of it, and then it would not be related to what your outcome is through anything but its more proximal effect, then you could use that to make causal claims. And he was doing this in the economics area, and they were looking at the effects of a duty on price and output with seasonal reference to butter and flaxseed. And what they used here was they were basically looking at supply and demand. Can you actually say what's driving things? Is it supply? Is it demand? And they used rainfall, which changed the actual changed yields, to say that this is changing the yields, which then allows you to know if the change is consequence on that it's likely to be due to the yield change and not to the demand change. So this was one of the first uses, or was the first. It was the first use formally when it was that was formally stated. Now, Philip Wright's it wasn't known for mathematics, and so it was often suspected that Sewall Wright had been involved in the development of this notion. So in the 1990s, I think it was, textual analysis was done of this appendix, and it was shown by textual analysis comparing it to Philip Wright's and Sewall Wright's writing that this was definitely written by Philip Wright. But of course, that didn't actually show where the ideas came from. And some of Sewall Wright's grandkids, or Philip Wright's grandkids, had the letters. And so the letters between Sewall and Philip Wright were found when it was absolutely absolutely clear that this was put together by the two of them. Philip Wright was asking, Sewall Wright for help. And Philip Wright said, I expect I'm stupid, but I don't seem to be able to pick up a new branch of mathematics so quickly as I could once. And they sent sort of attempted diagrams, and Sewall Wright was writing back. And it was clearly a collaborative effort. But Philip Wright said, I hope all this work you have put into this is not going to prove obsolete. I hope we should be able to see some results in practical application. And duly, a paper was submitted to the Quarterly Journal of Economics and was rejected. So no one knows who the referees are, but this meant that instrumental variables never came to never became known for 50 years, essentially, because no one read Appendix B of this this incredibly boring book on tariff and trade. And Sewall Wright never came back to it, which is interesting. It was, although it was mentioned in the 1940s, and this name instrumental variables was given but didn't take off. It took off when the famous economist Arthur Goldberger wrote a paper in Econometrica in 1972, where he actually called it instrumental variables. He used that name and he actually explained it, and he explained how it worked. And then it became popular, but largely in economics. It's hardly applied elsewhere. So now we've had the young Fisher, so now we get back to the old Fisher. And of course, Fisher was known as a brilliant statistician, founder of statistics, and also known as a brilliant geneticist. But he only once, to my knowledge, gave a talk where he combined his notions about statistics and his notions about genetics. And this was the Bateson Memorial Lecture given in 1951 on statistical methods in genetics. And here, which has been reprinted, nearly everything I talk about, I reprinted in the IJE, so you can find these things with lots of commentaries if you're interested in any of these things. That's the International Journal of Epidemiology. In that paper and that talk, Fisher said that genetics is indeed a peculiarly favoured condition in that Providence, with a capital P, he was a major believer in God, has shielded the geneticist from many of the difficulties of a reliably controlled comparison. The different genotypes possible from the same mating have been beautifully randomized by the meiotic process. Generally speaking, the geneticist, even if he foolishly wanted to, all of them he, of course, could not introduce systematic errors into the comparison of genotypes because for most of the relevant time, he had not recognized them. So, you couldn't fuck up the genetic study if it was done sort of properly because of the randomization inherent in meiosis. And then Fisher said about the connection between the two subjects, which was statistics and genetics. And of course, he has become famous for this introduction of the notion of randomization, randomized trials, factorial randomization after that in the agricultural sphere. And then Bradford Hill took that into medicine. So he says the connection between our two subjects, which seem not to be altogether accidental, namely that the factorial method of experimentation, now of lively concern so far afield as the psychologists or the industrial chemists, derives its structure and its name from the simultaneous inheritance of Mendelian factors. Geneticists certainly need not feel that the intellectual debt is all on one side. And when I reprinted that in the International Journal of Epidemiology, I asked his daughter, Joan Fisher Box, to write. She wrote this brilliant biography of Fisher. It's definitely a warts and all biography in 1978, and she wrote a 10-page. This brilliant essay. This is a brilliant essay about Fisher's Bateson lecture and how his work in statistical methods and genetics came together. And she said, in this, she just says unequivocally that the notion of factorial randomization in the agricultural studies came from the factorial nature of Mendelian inheritance. So Mendelian randomization came before randomized controlled trials in the scheme of things. So now I'm going to move on now when we can actually get germline genotype and use this for asking specific questions on phenotypic causality. And now I'm just moving into my sphere, which is, is thinking about human disease etiology in an epidemiological way, and how you can get evidence on whether something is truly causal, which of course is the major issue with epidemiology, when so many things that have been thought to be protective, like vitamin E supplementation or vitamin C or hormone replacement therapy or whatever, turn out not to be when randomized controlled trials are done, because of confounding, because of reverse causation, probably more important than it's generally considered, etc. So how can you use germline genotype to answer these specific questions? And this was used in, initially, this would be used in an informal way, but in a way that I think is an appropriate way, essentially, in the way of triangulating evidence, of taking evidence from observations and evidence from what you can get from genetics and evidence from trials, if you can do them, etc., and putting those evidence to get that evidence together. And if the evidence points in the same direction, you are more confident about the causal statements. And so very early use of this was the identification of familial hypercholesterolemia. There was, by 1910, around 1910, the notion that cholesterol might increase the risk of forms of heart disease, atherosclerosis and heart disease, was put forward, but it remained controversial for 70, 80 years. But it was pointed out that you have this genetic difference, which one could see being transmitted across humans, and that the genetic difference that went with higher cholesterol also translated into higher risk of coronary heart disease and other atherosclerotic disease. So this was used very, very much in establishing the idea that a higher circulating cholesterol level caused coronary heart disease. There's also some other uses in the historical literature. The identification of lactase deficiency relating to milk intake, just to a more modest degree than it was thought, and this was without genotyping, obviously, for lactase deficiency. This was thinking people were lactase deficient because they reported not liking milk. So it was a bit circular, but they thought that they were looking at something genetic, and therefore that people who were having low milk intake because they thought they were lactose intolerant had more osteoporosis, and so calcium deficiency was bad for you. So quite a lot of work was done on that without genotyping. Work was done in the cancer space when when polymorphisms, which were thought to either detoxify potential carcinogens or to activate potential carcinogens, were combined with observational data to see if you had an interaction between the genotype and exposure to, for example, cigarette smoking or alanine dyes, etc. And when it was shown that people with a particular genotype who also were exposed to what was thought to be carcinogenic, had a higher risk than people exposed to another, that was used as evidence that the phenotype influenced the outcome. It wasn't used to say, oh, look, there's this genotype which gives you high risk, or we've got to screen for this genotype. It was and stop people being exposed if they've got it. It was used to say, look, this phenotype is likely to this phenotype, like smoking or alanine dye exposure, is likely to increase the risk of bladder cancer across the board. This adds to the evidence that these things are causal with respect to this outcome. That's to be printed as well. And Jerry Lauer, who published that paper, just to say, his blog, this is 20 or 30 years after his paper, 30 years after his paper, says that these sorts of evidence doesn't imply that there are genes so intimately related to cancer that they deserve to be called cancer genes. It just helps you learn about how the environment that we can do something about influences the disease. So there's lots of those examples. But the first place that I know really where this was actually formalized up explicitly was a letter by Martin Catan in the Lancet in there's Martin in 1986, and he wrote this. That's in the IJE as well. Lots of commentaries. He wrote this at a time when there was a lot of controversy about whether low cholesterol level could increase the risk of cancer. Observationally, people with low cholesterol had high risk of cancer. I mean, but smoking reduces your cholesterol level. Infections can reduce your cholesterol level. If you start getting liver failure, it'll reduce your cholesterol level, and that, of course, can be an early sign of developing cancer. So you get reverse causation. There's lots of confounding, and so Martin just proposed that what you could do was look at ApoE genotype, which was then known to be related to higher cholesterol level and to a difference in cholesterol level, and say, look, if lower cholesterol actually has a biological effect that increases your risk of cancer, people who carry the genotype that's related to lower cholesterol should have increased risk of cancer. He didn't have any data. He just proposed it as a method, but he said your genotype doesn't change, so you won't get the reverse causality. So in the cancer, in this situation with cholesterol and cancer, it was reverse causation from early stages of the disease process that was considered important. And also that the genotypes wouldn't be confounded with things like smoking. The genotype related to cholesterol level wouldn't relate to smoking. It would be unlikely to be related to smoking. So it was that he put that forward as a proposal. It was cited twice between in 88 and 92, but entirely about ApoE biology, nothing to do with the method. But since 2003 has been cited highly. I'll skip reverse genetics. This is just to say that this notion of meiotic randomization was also used in animal type studies, looking at whether one brain region, the downstream effects of activity in one brain region on other brain regions. And this is meiotic randomization of hippocampal morphology, and the term genetic randomization was used. But the term Mendelian randomization was introduced in a context which is different to how it is used today by Richard Gray and Keith Wheatley in a truly ingenious proposal about how you could evaluate bone marrow transplantation in hematopoietic cancers, that's lymphomas, leukemias, in kids. Now, at this time, these cancers had a terrible outcome. The outcome was terrible on a diagnosis. No one would want their child to enter a randomized trial if there was something that might have any benefit, because the alternative was pretty grim. So trials would be very difficult to recruit to for understandable reasons. And so what Richard and Keith thought, they said, fortunately, however, the value of allogenic bone marrow transplant can be assessed unbiasedly without the need for formal trials by taking advantage of Mendelian randomization and comparing the survival of patients whose siblings are HLA compatible with that of patients whose siblings are not HLA compatible. And to me, this was a brilliant idea, because the notion here was that by far the biggest predictor of whether your child would have a successful bone marrow transplant, by far the strongest predictor was if they had a compatible sibling. Now, if they had a compatible sibling, would be due to Mendelian randomization. It would depend on genetic similarity, which would depend on how the die had fallen when the genotypes go from their parents to the offspring. Possibly the one issue that they didn't address was that you should take into account the number of siblings that the child had, but later studies actually then did match on the number of siblings, or studies that were then done did match on the number of siblings that the child had. But then, and so your analysis group would be like a trial. It'd be like an intention to treat analysis in a trial, because you would look at all of the kids with genetically compatible sibling, whether or not they got a transplant, versus all of the kids without a genetically compatible sibling, whether or not they got a transplant. But those two groups are just defined by the fall of the die when the genotypes were transmitted, whether they've got a genetically compatible sibling or not, if you match on the number of siblings, which should be done. And it'd be just like with a randomized trial, you analyze the people who have been randomized to the drug, whether or not they take it, called intention to treat analysis. You analyze the whole group who are randomized to get your statin. Even if 20% of them flush them down the toilet, you still include them in the treated group, and if 20% of them flush the placebos down the toilet, they're still included in the group. You don't use adherence as an indicator of whether they should be included in the analysis or not. Everyone's included. So this is like an intention to treat analysis, and the method has been successfully used in this area. Of course, it's very specific to this particular issue, although people have tried to think of ingenious ways of doing it. And we didn't cite this we cited Graham Wheatley, absolutely, in the 2003 paper that I wrote with Shah Ibrahim, which put forward the sort of rules of Mendelian randomization, as it now is, I guess. Because the one study that appeared by the time misspelt Mendelian randomization, or the spell checker got it and changed it to median randomization, so it wasn't found in any literature search, but these were done, and there's been, as I say, a fair number of such studies. And here's genetic randomization, and I first cited the Graham Wheatley paper just to use the term Mendelian randomization, but obviously in a very different context when thinking about homocysteine and coronary heart disease, which I was working on in the late 1990s, early 2000, and said that it was impossible to get any sensible answer from observational studies because homocysteine is just so strongly confounded by everything. Everything bad puts up your homocysteine. And disease, there's reverse causality. If your kidneys are failing, your homocysteine goes up. So of course it predicts mortality. You can't adjust for that statistically. You just simply cannot adjust for a wide range of confounders and reverse cause. But there was genotype related to higher homocysteine that you could use that to get evidence on this issue. And David Clayton and Paul McKeague discussed this, and Bernard Keaveney, these are sort of very early papers discussing this approach. So the key concepts in 2003. So the key concept of Mendelian randomization, as Shah and I saw it in 2003, when introducing this, was that we wanted to use this approach to say, could you actually improve human health? Does it give you evidence on how you could improve human health by manipulating something, like by lowering homocysteine level or lowering cholesterol level? Or was it worthwhile increasing HDL, or good cholesterol level as it was called then? The answer to the latter one is no, by the way, which Mendelian randomization would have saved billions of quid if people had actually used the already emerging data that showed that genotypes related to HDL cholesterol don't relate to lower coronary heart disease risk. Quite a lot of money would have been saved. But anyway, so the fundamental principle was of gene environment equivalence, by which we meant that if your homocysteine is higher because of environmental factors, or if it's higher because of genetic factors, it would have the same downstream effects on your outcome. It doesn't matter how you manipulate your homocysteine, whether it's genetic influence or whether it's environmental influences, the downstream effects is the same. So that's quite a strong assumption. It's an assumption which in medicine there's quite a lot of examples to support it. For example, with Hartnupp's disease, which looks like pellagra, has the same symptoms as pellagra, and it's a genetic disorder resulting from poor absorption of amino acids, including tryptophan, which is converted to niacin. Pellagra is due to niacin deficiency. I mean, pellagra was shown to be related to dietary deficiency, and just brilliant work by Goldberger in the 1910s, 1920s but they didn't actually identify the factor. Niacin hadn't been identified till decades and decades later. But if Goldberger, if had known about, if Hartmut's disease had been understood and people had known about how it worked, he would have jumped straight to niacin. So, so you consider this to be like, phenocopy and genocopy, which sort of mean the same thing. It just depends which way you're looking, which direction you're looking. That the environment, phenotypes can influence phenotypes, and genocopy is that you can mimic something caused by phenotypes with a genotype. So so there's in some developmental genetics, people like Mary Jane West Eberhardt refer to gene environment equivalence, gene environment interchangeability. It doesn't matter how gene expressions changed, it will have the same downstream effects. Well, I think Zuckerkandl and Willett possibly went too far when they said no doubt all environmental effects can be mimicked by one or several mutations. I think being hit by a truck, difficult to think how that can be mimicked. But I think most forms of endogenous biology which influence diseases are so mimicable. This another example, a recent example in COVID-19 is where genetic variants influencing IF and it relates to severity of COVID. So people who have a deficiency, inborn errors of interferon, of this interferon component, had high risk of dying of COVID, and so did people who had autoantibodies. So 4% of people, increasing percentage as you get older, of people have circulating autoantibodies. They don't have any genetic influence on they don't have an inborn error in the gene producing this interferon component. They've developed, phenotypically for some reason, they've developed circulating autoantibodies, which of course reduce the biological activity. And the two have the same impact on the disease outcome. So Mendelian randomization links strictly to Mendelian principles. So we introduced Mendelian randomization as being using the randomization of genotypes from parents to offspring. That is the true randomization. And we said that that was analogous to a randomized trial because there was true randomization. And we said, and we discussed how this relates to Mendel's laws. So it relates to the first law in that the segregation through to the person entering your study. So the segregation has to be still random at birth. So I mean, you could have, meiotic drive, or you could have some form of fetal selection. That by environment, mothers who drink very heavily might abort kids with MTHFR variant, for example. So there are possibilities of subversion of this, but so the assumption has to be that randomization occurs at the development of gametes and carries on down to when the people get into your study as the first, and then the second, of course, is that beyond things that are in close physical proximity in the genome, that the genetic variants travel independently. And of course, at the time we were writing in 2003, there was no possibility of doing any Mendelian randomization study with parent offspring data because there just simply wasn't enough data. In fact, there was one that could be done. MTHFR in the mothers could be shown to increase neural tube defects in the kids. In fact, that was the one example of parent offspring Mendelian randomization that did exist or was possible at that time, and then said that low folate, that basically said that low folate in mothers increased the risk of neural tube defects, which it does. And of course, there's been trials on that, and even the UK is now doing supplementation with folate to a small degree to try and produce that benefit. But there was essentially no data. Data just didn't exist, and secondly, you couldn't test these. You couldn't test the truth of the Mendelian laws in humans. There was ridiculous papers with tiny sample sizes claiming that this genotype shows that it can overtransmit, but most of those didn't replicate. I mean, there are examples of selfish elements in other species, and some discussed in humans. But now, now the Norwegian mother and child cohort has 100,000 parent offspring trios. So we are hoping to produce some sort of map of the extent to which, and you've got DNA at birth to see the extent to which there might be segregation ratio distortion, transmission ratio distortion during the fetal stage, for example. But in 2003, it was not dreamable of. And so we said that in that situation, then what the best that could be done was what we called approximate, rather than absolute Mendelian randomization, which was using population genotypes and using the approaches that have obviously developed since then, of using principal components, etc., as ways of taking into account population structure. And you could use, using those ways, but the degree of certainty of the randomization is less. So in conventional observational studies, as I say, you take an exposure like cholesterol level, you see how it relates to coronary heart disease, but there could be confounding and reverse causation. Your instrumental variable would be a genetic variant related to cholesterol level. There would be no reverse causation. Early coronary heart disease couldn't influence your cholesterol level, couldn't influence your genotype, even though it could influence your cholesterol level, no confounding, and therefore the joint association of your genetic variant with the outcome with coronary heart disease and your genetic variant with the exposure, cholesterol, allows you to make some estimate of the lifetime effect of the difference of cholesterol level on the outcome, which is basically done by dividing the genetic variant outcome association by the genetic variant exposure association. That's the Wald ratio in an IV analysis. And the gene environment equivalence idea is just the notion that if diet or cholesterol lowering medication influences your cholesterol level, it has the same downstream effect as if the genotypes do. In this particular situation, that's very reasonable, because amongst the genotypes are HMG-CoA, which is the target of statins, NPC1L1, which is the target of ezetimibe, PCSK9, which is the target of PCSK9 inhibitors, etc., etc. There are genetic mimics of all the drugs, and the trial data and the MR data match up extremely well. I'll skip that. So the instrumental, this is what Sewall Wright and Philip Wright had introduced, and the assumptions now that are discussed at length is that your instrument genetic variant has to relate to your exposure very reliably. There must be no confounding of your instrumental variable, your genetic variable, and the outcome, and the confounding would be population structure, but could also be dynastic effects. If the genetic variant in your parents, say genetic variant related to educational attainment, influence the environment that your kids are brought up in, then the genetic variants become associated with those factors. So confounding can be introduced. So that's why you need within family studies. If you do between sibling study, of course, that can't exist. So that's what, so now we have massive sample sizes and you can do between sibling studies to take that into account. And the most challenging assumption is that the genetic variant is independent of the outcome, except the only effect of the genetic variant on the outcome is through the exposure. And of course, pleiotropy or horizontal pleiotropy can bias that. So we have a historically informed way. What possibilities does that give? So back to T.H. Morgan. Morgan introduced what we now know as the omnigenic model in 1917, so that the whole germplasm, across the whole germplasm, it's producing effects on everything. And the omnigenic model is just if you know this work, is just a Mendelian randomization model, multivariable Mendelian randomization model, which says that the outcome, that the effects of the what we'll call the core genes, it's regulation of core genes by upstream genetic variants which regulate those genes is produced through activation of those genes. You can use Mendelian randomization on fitness if you know what the fitness is. So in a case-controlled genetic association study, selection to the case group, getting into the coronary heart disease group drags along genotypes related to higher cholesterol level, for example, which is why in that case control study, you have more genetic case control study, you have more genotypes related to cholesterol level than you do in the control group, which gives you the MR result that cholesterol causes coronary heart disease. But in evolution, selection is by phenotype, and the phenotype drags the associated genotype to an increased prevalence in the future generations. And gene environment equivalence is, by the way, assumed by the breeder's equation, because you say the selection differential is the covariance of the relative fitness and the trait value. And then this shows that when you then multiply by the selection differential by the heritability, means you're saying that the stuff which isn't heritable in the phenotype has the same effect. So gene environment equivalence is assumed in the breeder's equation. The analogy between MR and artificial selection is, of course, seen in experimental studies, and is now, Mendelian randomization is actually now being used under the name Mendelian randomization in the agricultural field. Pleiotropy exists in Mendelian randomization as a major problem. There are about 27 different approaches to doing sensitivity analyses for MR to allow relaxation of these assumptions. But of course, you see that also in natural selection. The famous farm fox experiment where these foxes were selected and bred from who were tame, and they developed sort of nice fluffy tails. It was thought that the fluffy tail wasn't been selected on, but if you just looked at phenotype, there are many phenotypes which change. So finding the phenotype is what's difficult. And basically using something equivalent to MR has been proposed for reducing environmental bias. But I've got 1 minute left, so I'm going to skip that. But so it has been proposed, and very recently, one seeing in studies of selection, seeing attempts to use these sort of path analysis driven methods. So there's a problem of fitness as a phenotype in humans. That problem is sort of taken away in one way and then reintroduced if you actually just look at whether something's been selected for. So this is from a paper by Ian Matheson, and this is the rise of lactase persistence. The alleles rising, the lactase persistence alleles rising very quickly and rapidly, suggesting there is some quite strong selection. And it's then said that it's the ability to consume milk that is driving this is proposed. But if you go back and look at the data now, we can genotype. It's evident that the lactase persistence genotype, at least now, relates very weakly to milk consumption. Tiny difference in actual milk consumption by lactase persistence genotype, making it unlikely, unless something has changed dramatically, changing that it's the ability to use milk that drives this. But in principle, you could use Mendelian randomization approaches to try and look at what phenotype it was, but you'd have to think back and get some data and think about when the selection occurred, or FAD's genotype increasing dramatically too. I mean, these are related to diet and processing of diet. So, so the issue, of course, is the pleiotropic effects of the alleles, and as I say, Mendelian randomization approaches could help. So I'll finish with, Sewall Wright really, thought physiological genetics, how genes lead to change of phenotypes. That's what he was interested in. He was also interested in ecology of populations and also interested in natural selection. And in a way, he introduced the methods of taking this apart, of taking those things apart. But he said, from a more general standpoint, all of this is merely an elaboration in terms of modern genetics of the conception of evolution by natural selection advanced by Darwin in the origins of the species. So we've gone full circle back to with phenotypic causality. And with genetics, the four concluding things to think of is, firstly, what genetics have shown us is the difference is that the often relatively small proportion of variance that is explained by tangible factors we can measure. And this is also shown by genetic studies using clonal species and trying to see how much you can constrain life expectancy in clonal C. elegans, for example? The answer is very little by trying to make the absolutely standardizing the environment. So this notion of intangible variance is something which, as I say, is closely related to non-shared environment. Is something that puts constraints on what we think you can explain. You do have strength and causal inference using MR, although I know quite a lot of people, some people around here don't like this. I think you can. I think it could be applied to fitness as a latent phenotype. But as with all causal inference, you should use triangulation of evidence principles. And sadly, this was supposed to come out before Christmas, but because of COVID, Cold Spring Harbor Press took ages to produce it. But there's a book on combining human genetics and causal inference to understand human disease and development, which would have been the perfect Christmas present for all your friends, but sadly, you'll have to wait till next Christmas. Thanks very much. I went on way too long. I know most people have to go. Sorry.
Well, thank you very much. Do we have any questions?
Email me questions.
That too, yeah.
Thanks a lot. My question is about this intangible or chance, because I think in most applied modern epidemiology, we don't have that. We don't like it properly. We just call it error. We can think we can connect it better.
Yeah.
Eventually get there and say everything. And equally, I mean, it's also there the genetic side of things, environment factors, but in terms of genetics, we're still far from really understanding the gap between, heritability.
Yeah.
What is chance?
Yeah, no, I think that's a really good question.
George, would you mind repeating it for the people at home, please?
Yeah, the question was, what is chance in the intangible environment, the intangible variance? And so I think that it will very much depend on the particular phenotype you're looking at. I think there's part of the chance will be things like early mutations, early somatic mutations, early epi mutations, or just a lot of modeling has been done on the notion of cytoplasmic division not being 100% at the early stages of cell division, and how those very small effects can have downstream developmental effects. But also I think chance can be a biographical aspect of people's lives. I mean, example sort of thought experiment I often give is it's a picture of Winnie, who was appeared in the Sun newspaper because she got to 100 years old and she's lighting a fag of her 100th of the candles on her 100th birthday cake. And then, of course, and it says she's not stopping now, and we all want to understand what's Winnie got that we haven't got, that people are dying early of smoking, and of course the tobacco companies 30 years ago that they really wanted people to find the genes which made people susceptible to smoking, so you could just flog fags to people who weren't susceptible. But more plausibly is because we know nothing's ever found. So there has to be a Winnie, there always has to be a tail of the distribution through just events that have happened right the way across life. So let's imagine that when she was 60, Winnie's at home and the postman knocks on the door at exactly 11 a.m. and she opens the door, gust of wind comes in, she coughs up a metaplastic cell that would have become a cancer. If the postman had come at 10:59 or 11:01, Winnie wouldn't have been around to smoke to have her 100th birthday. If we'd rolled back the time and changed the postman, Winnie would have been gone. But if we'd rolled back the time and changed people's lives, some other person would then have come through to fill that spot of the people who makes it to 100. So those things are environmental, but it's just impossible to think we could ever know those factors. And I mean, you can think the same of, for example, ubiquitous exposures everyone's exposed to, like Epstein-Barr virus. 95% of people show evidence of being infected by Epstein-Barr virus by the time they get to 25. Every single case of multiple sclerosis the person's been infected. So Epstein multiple sclerosis doesn't occur without Epstein-Barr virus. But that's not very useful in terms of thinking about it, because the exposure is so common. But it's probably timing. It might well be timing. If you get Epstein-Barr virus late, you're more likely to get symptoms of infectious mononucleosis, glandular fever, and like all late virus infections have generally much worse effects. If you get yellow fever at age 2, not so bad. If you get yellow fever at age 25, it's absolutely terrible. So it could be the timing of the infection. Now, Epstein-Barr virus is one of the few cases where you can actually demonstrate that, and in fact, trials are, I think, being started of Epstein-Barr vaccination in people at very high risk of multiple sclerosis to try and do something about that. But imagine if that effect is a maternal, if you're interested in neurodevelopmental outcomes, imagine if that is a maternal infection, a ubiquitous infection that just everyone gets, circulating around, and if you get it at 5 weeks, it has massive developmental consequences. If you get it at 6 weeks or 4 weeks, not. Now, I mean, we have an example of German measles where we can actually where that was actually identified. But imagine if those sorts of things are incredibly difficult to identify. Ubiquitous exposures where essentially everyone's exposed in, but at different times in places are incredibly difficult to identify. So I think that, I think there's certainly a component of biological stochasticity, developmental mutations, etc., in the process, but also there might be a very substantial amount of just environmental stochasticity, just the sorts of things that we just can't measure. It's depressing, but I'm heading for retirement, so that's okay.
Any other questions?
I'm very happy to take questions by email later if anyone's anyone comes up with anything. And I'm sorry I went on so long.
Thank you very much for your talk.
I shouldn't.